Yes, we can see and hear you. And just for everybody, we are recording this session and it will be available later as a video.
Super. And I get to start, which is my great pleasure. It's really nice to see so many of you, and it's really nice to see so many people whom I know from before, and so many people whom I don't know from before. And I think that's part of the pleasure with this overall project. And the reason why we wanted to organize this webinar exactly now is because now we're actually all really fully set to get going. We have sent out the material. We have teams eagerly waiting to start sampling. It's all in place, and it's just the time to get going. So what we thought we would do today is to have me. I'm the PI of Lifeplan from Sweden. Talk about why there is a Lifeplan in the first place. Then we'll have Bess talk about how this whole thing actually works. Then we'll have Dee, another coordinator from Sweden, talking about the logistics and lab work in Sweden. And then we'll have Otso, another Lifeplan PI, talking about what we have done and will be doing with the big data collected in the project. And then we'll have two presentations from actual teams contributing to the project, so that you get to see some real people involved here. And of course, the idea is also that you get to see each other, at least per TV, which is very important. And then at the end, you get to ask questions in the chat, as Bess was saying. But let's start with the whole background, like why there is a Lifeplan in the first place. And I guess that question goes back to the reason why we are all on this call, because I'm convinced that absolutely everybody who has signed up and who is here today, we all share the same passion, and that's the passion for life and for diversity on this very planet. And what we all work on is figuring out how that all of that life is patterned across the globe. And I think we also share a common dream of making this science of ours and actually a predictive science, so that in the best of all possible worlds, what we will soon-ish be able to do is to point to any kind of place on the planet and to ask, given the conditions at this site, what is the likely state of biodiversity exactly here? And if we reach that understanding, then we will also be able to predict how will this diversity change, given, unfortunately, mostly human-induced environmental change. So this is the level of understanding that we're all aiming for together. And this is what Lifeplan is aiming for. And the reason why we felt that this project is ripe for happening exactly right now is because there are several long-term developments which are coming together exactly right now. And I will run you through them because they form the basis for the whole project. And those are really the new technological developments which finally allow us to describe biodiversity in an efficient and standardized and comparable way. But there's also the emergence of simply new ways of doing science and doing so together, as is also illustrated by the fact that we are now 107 people on this call. And the third thread entering this overall constellation is the rise of the development of general ideas about how diversity works, how communities actually come together. So new ways of generating data, but also new theory for understanding that data. And Lifeplan is about bringing all of these three threads together. Now, the first one, the new methods for characterizing biodiversity. Well, we all know that biodiversity is essentially, well, it is exactly just that, diversity, or a mess. And making sense of that has been tremendously hard through time. And now during the very last 10 years, I would argue, we're finally seeing the methods allowing us to make good sense of the major parts of biodiversity, not just the particularly species poor groups. And we will be drawing on all of these developments, well, not all, but all of the ones shown in this picture. But it's not only about technology, it's also about new ways of making science. Because what we have also seen during the very last few decades is the emergence of new ideas for collaboration. And I guess as a community, we finally understood that we don't have to do everything as individual groups, sending people around the globe and getting frustrated. Instead, we have found new ways of actually teaming up as a community and sharing the workload, so that we simply agree on how to do it, and then we all do it more or less at home. And that way we build the big picture. And I think this is extremely stimulating to do science together like this. And the third thread and development leading up to Lifeplan, that's really the development of general theory in community ecology. Because if you think about it, I mean, community ecology in a sense has been there for several centuries, but it mostly started with just like people were describing species, they were also describing communities and community types, vegetation types. So a classical typology. And what has happened then very recently, or well, during the last half century, I would say, is the development of ideas, of models of how these patterns come about the processes actually forming them. And that development has been very, very quick. And the final step needed, which I think we are seeing exactly right now, is the emergence of efficient techniques for uniting patterns and processes, for finally kind of distilling the imprints of these processes from real data on communities. So I think Lifeplan is about bringing together these exact developments, because what we are doing here is, this is a map of all of you across the world. And what we are doing together is to do science in a new way, doing it from our respective sites, from our respective research areas, teaming up, applying these new efficient techniques for characterizing biodiversity, converting them to actual records of what species occur where, and then distilling the imprints of processes, of ideas of how these communities are formed from the data, and using that to revise joint theory of biodiversity. So this is as I see it, and as I hope you see it, the actual basis and outline of Lifeplan. And with that, we shall turn to Bess, who talks about how it's actually supposed to work.
Thank you. I'm going to share my screen here. You seeing that okay?
Yes.
Great. So how does Lifeplan work, and is it actually working? The sampling design that we have is trying to do two things. It's trying to contrast different spatial scales, and it's trying to contrast natural and urban environments. So at the global scale, we have about 100 sites all over the world that alternate between a natural and urban location. And at the national scale, we have higher densities of sites in the Nordics and in Madagascar, with the sites not moving but always staying at the natural location. And then at the hierarchical scale, we have this nested grid. We're going to do one grid in Sweden and one grid in Madagascar. And this nested design gives us distances of 50 kilometers, 5 kilometers, or half a kilometer between plots. And what is a plot? It's 100 meter by 100 meters square with all 5 sampling methods in the center: cyclone, malaise, soil, image, and audio. And then additional image, audio, and soil sampling at the 4 corners. And from these 5 methods, we get 3 kinds of data: DNA sequence data, image data, and audio data. Over the course of the project, we expect to collect about 5 petabytes of data and the associated metadata that tells us where and when each piece of data is from. And this diagram, which is a bit crazy, summarizes how the data in purple and the metadata in blue gets from the bottom left box, which is you, the teams all around the world, to the box on the right, which is the data center at Finland's Center for Scientific Computing. So to start with the metadata in blue, you collect it by scanning QR codes on the traps and samples with a smartphone app, and the app collects it in the metadata table here. For the actual data in purple, you collect it to start with the digital side here on the right, you upload it to Nextcloud and make a local backup. And then the software called the middleware moves it from the Nextcloud to this large data repository, and at the same time connects it with the metadata. And the way the metadata and metadata actually get connected is here by you, the teams, in this gray arrow where you do this manual copy paste of a share link from Nextcloud. That's where the actual magic happens. For the physical samples, the soil and spore samples you send to SLU at Uppsala for processing, pre-processing, and the Malay samples you send directly to Canada, to Guelph. And from Guelph, we get DNA sequence data, which is digital data, and they will send it to us by much the same system as the rest of the data. So what we end up with is 5 petabytes of sequence, image, and audio data, mostly audio data, where we know where and when each piece of data is from. So is it working? How are we doing? To start sampling, you need to have 4 things in place. The first is the cyclone sampler, which got sent from Burkhard in the UK. If you haven't received yours, then we may still be waiting for a delivery address or some other paperwork. So 89 out of 116 teams have received theirs. You also need the rest of equipment, which gets sent from Sweden. 73 of you have received that. If you haven't, then please confirm the shipping list that Deirdre has sent. Then there's a collaboration agreement, one that details the collaboration agreement tells who is responsible for what and who pays for what. And 60 of you have signed it so far. You might still need to refer to a draft or to the PDF sent for your signature. The slowest one has been the material transfer agreement, the MTA. So this is the one that details ownership issues and access and benefit sharing in accordance with the Nagoya Protocol. This is the slowest process because countries differ in requirements, so may not even have a draft of it yet, depending on what country you're in. But the signing process, once you have a draft, is much the same as for the CA. So 35 only up to 38. And this is what it looks like on the map. The red teams are still missing some piece of the puzzle. Yellow teams have all the pieces and could start sampling. And green teams have started sampling to the extent that we've received audio image data. How is the data flow working? It is working So the app is available. There's going to be an update within a week with improved QR code scanning. So you should then be able to scan even those annoying tiny QR. The Nextcloud has had a lot of problems in the outset, but disk space has now been increased and it's working now. The middleware is working. The next thing to figure out is the physical sampling. We'll see how that's, but it hasn't started yet. So all in all, let's go. And the next presenter is Deirdre.
So hi, can you hear me and see my screen?
Yep.
Great. So we're based in Uppsala in Sweden at the Swedish University of Agricultural Sciences, and we're a team that's in charge of the field equipment, some of the lab work, and also some of the field work for the nested sites. So to start off, who are we? Well, there's 4 of us. There are 3 research assistants, Bianca, Gaia, and Hannah, and then myself, Deirdre, the other coordinator. And with Bess, we have been trying to answer your questions on Slack or from the emails that you sent through the Lifeplan email. So you may have heard or seen us already. And also, last but not least, we've been responsible for sending you your kit. This has been trial and error and a lot of work. We've been working on this since last April. It has been acquiring a lot of equipment, testing equipment, counting equipment, repacking it, reprogramming it, recounting, and eventually, once you've approved your packing list, we have been able to send it to you. So as Bess mentioned earlier, at the moment, we have managed to send the kits to 126 sites to 32 different countries, and that's about 2.5 tons of equipment sent so far. This includes 53 sites for the hierarchical sites in Madagascar, and we have a few more to go, but we're way over halfway, so we're pretty excited about that. And then, when we're not in the basement packing boxes, we have been preparing for the lab work. So we will be the ones responsible for receiving and processing the spore samples from the cyclone sampler, as well as the soil samples. So I've done a rough estimate, and we're going to generate about 40,000 spore samples over the duration of the study and about 30,000 soil samples. So each sample gets sorted a bit differently. So I'll start with the spore samples. When we receive them, we will carefully remove any insects or dirt, and then we will freeze dry them and send them for DNA extraction and sequencing to the Center for Biodiversity and Genomics in Canada. When we receive the soil samples, then we freeze dry them, and then we are also the ones that will be doing the DNA extraction and processing before sending them off to sequencing. And on top of that, when we're not in the lab or in the basement, we have been working on setting up the 28 hierarchical nested plots that will be around Uppsala. So we will be visiting these sites weekly. And yet, once again, we've estimated that's about 1,260 AA batteries to change per week and 280 SD cards. Yeah, so we're really excited that some of the sites have started sampling and that we're working with all of you. Thanks for listening. And I think next.
Okay, hello everybody. I hope you can hear me okay and see my screen. So I will talk a little bit about the Lifeplan data analysis pipeline. So what happens after the data are collected and sequenced and processed? And let me first remind you that Lifeplan really combines biodiversity research with development of statistical methods. So David Dunson is one of our PIs who is not here today, but he is really an expert in big data statistics per se, without specifically related to ecology, but in terms of fundamental statistics. So in this project, we combine biodiversity research, where Tomas is the leading PI, with the development of fundamental statistical methods, where David is the main PI, whereas my role as a kind of a statistical ecologist is to provide the common language between these two fields. So roughly speaking, in a very simplified way, our data analysis pipeline consists of these steps illustrated in this slide. So the first step with the DNA sequences, images, and audios is basically to identify which species are present in each of the samples. And here, of course, we need automated species identification methods because we have too much data for any expert to go through it. Then the next step is to use these data to provide an improved understanding of biodiversity dynamics. So here, one method that we will be applying is joint species distribution modeling, which is something that we have been developing ourselves. And then as a third step, we want to relate our empirical results to spatial theory of community ecology to provide kind of the broader context. So in terms of the kind of the methods fields, we are talking about bioinformatics, statistics, and mathematics. I cannot go through all of that in a very short time, but I want to give you one example of species identification challenge and then one example of the ecological inference. So let me first say a little bit about how we plan to identify global birds from the audio data. So here I illustrated 1 minute segment of audio data. So you see here it time versus frequency. These tiny lines here are some birds vocalizing, as I have circled them by the rectangles. And now the challenge here is that we will have of the order of 1 billion of these kind of 1 minute segments. So nobody can really listen through them manually. We need to identify the birds with the machine learning methods. So the main idea is to combine citizen science with machine learning, basically to teach the computer identify the birds of the world. So in addition to this distributed sampling that you are now doing, we plan to have a distributed annotation campaign to basically learn about the classification features. So we are utilizing existing databases such as the fantastic Macalau library, but we also plan to run our own citizen science projects to complement these as needed. And this is actually something that we have already started in Finland. We recently started a pilot project where we created together with Luomus this web portal that you see illustrated here. The text is in Finnish because this is targeted to Finnish audience. And here the bird experts are invited to identify the birds from short samples of sound. So we generate these samples in a way that they are as useful as possible for training our models. And now we invite the expert to tell which birds vocalize in which segments. So the next step is really creating a similar web portal for generating training data for global bird identification models. And once we have this up and running, the idea is to invite all of you, those of you who know birds, to take part to this process, basically to help us to teach the computer to identify the birds from your very own samples. Then I wanted to give one example how we are learning about ecology from Lifeplan data. Well, we don't have the Lifeplan data yet, but we have pilot data such as the GSSP data, the global spore sampling data that was mentioned already. So recently, we finally got all the samples of these data sequenced. We have something like 2,100 samples from 47 locations. The sequences total some 100 million fungal ITS sequences, with the estimated number of roughly 300,000 species of fungi in these, of which we can identify only a tiny fraction to species level in a reliable way, but about half to genus level. So here in the map, you see the GSSP sampling locations in black, and actually we are comparing these to the earlier published soil sample data by Leho Tederso in 2014, so also those sampling locations are shown in this map. So what can we learn from this kind of data? One thing that we learned is that fungal communities seem to be structured very strongly by climate, but not really by distance. So here in the ordination space, you will see the sampling locations, so that locations nearby each other in this ordination space, they have similar fungal communities. And you see that, for example, these sites that I have circled here, they have similar fungal communities by large. They are close to each other in the ordination space. They are all over the place. One in South America, one in South Africa, one in Australia, one in Europe. In spite of this huge difference in distance, they have really similar fungal communities because they have similar climate. So this kind of supports the notion of everything is everywhere, environment selects, not so much the dispersal limitation at this scale. Another thing that we can learn from this data is that there is very strong seasonality in fungi. So that, of course, in the temperate and in the Arctic regions, the amount of fungal DNA that there is in the air, how many nanograms of DNA you have per cubic meter of air, that really peaks heavily in the summer season. But what is quite interesting is that it basically never exceeds that we see in the tropical regions, that in the tropical regions you have really high DNA abundance of fungi in the air. And the last thing I wanted to say is that we have used the pilot data to estimate how much one should sample globally to represent basically global biodiversity, especially for insects and fungi, really the unknown groups. And what we see from these estimates is that with the Lifeplan sampling design, we will not cover every single species in the planet, all of the unknown fungi and insects, but we will really get a good proportion of them, maybe up to half. So just to conclude, I remind that we are really excited not only to collect the biodiversity data, but also at the same time develop new methods of bioinformatics, statistics, and machine learning to better interpret this data. And so it will be really exciting to see where we get after a couple of years of data collection. Okay, thank you very much.
Thank you, Otso. So next we will see some short video introductions from teams and sites in Perth and Termoli. I'm going to share them here on my screen, but because sometimes sharing videos works a little bit badly, I'm also going to post links in the chat so that you, if the sound or video doesn't work for you here, you can go and watch them on YouTube, so we can all watch together. But I'm going to try and share them with you here. So I'm going to play them back to back. First the video from Perth and then from Termoli. Let's go full screen on that.
Okay, hello. My name is Adrian Pinder, and I'm with the Department of Biodiversity, Conservation and Attractions, which is the Western Australian Government Conservation Agency. I'd like to thank Lifeplan for giving us this opportunity to introduce the Perth site. And the first thing I'm going to do is introduce our team. So the three team leaders are myself, Steve Easton, who is a botanist from the Botanic Gardens and Parks Authority, and Jeff Barrett from our Swan Coastal Plain office, and a keen birder. So Jeff will be helping us with the training data set for the audio data. Now, the people who will actually do most of the on-ground work and the data uploading are Adrian Barrett and Kirsty Quinlan, who will manage the natural site, and Martin Brotherson and Ryan Glowicki, who will manage the urban site. So this is Australia, and in Perth, we're in the far southwest corner of the continent, just here. And we're one of the most isolated cities in the world. So zooming in further, this is Perth City here, and you can just see the urban sprawl. And there's a distinct line here that you can see. Now that's an escarpment that separates a largely cleared coastal plain just here from an elevated hardwood forest. And then east of the forest is a vast area of dryland agriculture known as the wheat belt. but we're focused on this strong coastal plain area near Perth. So this shows the locations of our two sites. Our urban site is Kings Park, which is an area of bushland very close to the city centre, but in the middle of the city. And our natural site is Lowlands Nature Reserve, which is one of the largest areas of remnant woodland close to the city centre. We have a Mediterranean climate with cool, wet winters and hot, dry summers. And we have an annual average rainfall of about 730 millimetres, but this is falling rapidly. We're certainly at the pointy end of climate change in southwestern Australia. So, starting with our lowlands site, this is an area of open woodland dominated by Banksia in the family Proteaceae and Alocasuarina, with occasional taller eucalypts and a variety of understory species. And you can see from this image that the nature reserve is surrounded by grazed pasture lands, and passing through the middle of the reserve is the Serpentine River. So this photograph on the bottom left here shows you what the site is like on the ground. So low open woodlands. The ground looks pretty bare in this photograph because it was taken in late summer. In winter there'd be more ground cover and the whole site would be greener. So late winter and spring is our plant growth seasons here. And then I've just added in a couple of photographs of the site installation, which was about 5 weeks ago. And then this top right photograph shows our first photograph of a native mammal captured using the Lifeplan cameras. So this is a brush-tailed phascogale just here. And I can show you what that actually looks like. So it's this little guy. Now, I've also mentioned some of the threats here. Introduced predators, so cats and European foxes are a really big problem here for our native vertebrates. And they would certainly be preying on things like this phascogale. And we have seen a fox already in the photographs that we've collected from this site. I've mentioned the fact that we're experiencing pretty acute problems with declining rainfall, and another big threat here is Phytophthora fungus, which is a soil-borne fungus, affecting many of the plant species that we get in these sorts of woodlands, particularly the Banksias. And it's not that fungus is not in this site yet, but it is known from quite close by. So we have quarantine procedures that we put in place as we enter this site, and hopefully we don't pick up Phytophthora in the soil samples that we collect over the next few years. Okay, so that's our natural site. The urban site, so this is Kings Park here on the banks of the Swan River. And this is the city centre here. So you can see how close this site is to the city centre. It's certainly within an urban matrix. This top right photograph here was taken from Kings Park overlooking the city and the river. And then I've included a couple of photographs of what the bushland looks like in this area. So it's a very similar structure and composition to our natural site. The threats to this site are very similar as well, with the added pressures of the site being at the urban interface. So more people visiting the sites, weeds coming in from gardens, and there not being the broader matrix of natural environments around it. Okay, so that's about it. I'm just going to finish off with a short video that I took at our natural site just this morning. Okay. Okay, thank you very much. I look forward to working with lots of you over the next 6 years on this exciting project. Bye.
Good afternoon. My name is Andrea Sciarretta. I'm one of the scientific team from University of Molise. Here we are in the Termoli natural site. That is the place where we are going to monitor biodiversity for this year. The next year, this site is 15 kilometers north of the town of Termoli, along the Adriatic coast in Italy. Next year, we will go to the town of Termoli for the urban site that will be into the town. In addition to me, the scientific team is composed by Angela Stanishi, that is botanist, Maria Laura Carranza, she is vegetal and ecologist, Mirko Di Febraro is an animal ecologist, and we have also other collaborators that assist us in the technical operations, that are Tania Travaglini, Marco Colacci, Francesco Tozzi, and we have 2 technical staff from the regional forestry office also that participated to the field activities. The plot, the natural site plot here has a rectangular shape. It is more or less 120 meters long and 80 meters wide. We placed 5 monitoring points, 4 at the corners and 1 in the middle of the plot. The natural site of Termoli is placed in the inner coastal dunes of Italian Adriatic coast, inside the Nature 2000 Special Area Conservation. The landscape is dominated by Mediterranean maquis and garrigues, and patches of open Aleppo pine and holm oak wood, with several eucalyptus trees planted over 30 years ago. The sampling began on the 1st of February. Now I show you some of the highlights of the survey we are going today. The cyclone was placed on a platform and under a small canopy. This is in progress. It is the place procedure. Here the vial is inserted. The battery is protected from rain and wind by an upside-down plastic jar. Here you can see the Malay trap positioned at the transition point between the forest and the more open bush. This is the collect procedure. Not many insects this week. We expect to collect samples year-round. Here is the QR code being acquired. Now the new jar with alcohol is being placed. This is the collection procedure with the camera trapping. Extraction of the SD card, changing batteries, scanning the QR codes of the camera and the SD card. Here, camera is reactivated with a new S card. And finally, audio recording. Also here, collection procedure. Scan the SD card. This is the most delicate operation because of the small size of the QR code. Then the activation of the audio recording with the audio moth application. And the installation of the audio recording in its case. This ends up our presentation. Many greetings from the Unimol team and best wishes for the activities on your side.
Okay, thank you so much to Team Perth and Team Termoli for putting together some really great presentations on a very short schedule. And next we're going to go into the question and answer session chaired by Otso Ovaskainen.
Yeah, I also want to say that those videos were really great to see. So thanks very much for making those. So now we have about 20 minutes time for questions and answers. And the idea is that you would mainly ask those questions in the chat, and then all of us are answering the questions at the chat as they are coming. And then I might highlight some of the questions here by speaking, and also maybe direct some of the questions to the members of the team. So I propose that everybody opens the chat as a big screen, and then you just start writing your questions or answers. And during those introductory presentations, there were some questions that were already answered about, for example, whether we do only birds from the audio data, but how about the other groups? And I see that Petteri answered already that yes, also bats and possibly amphibians and mammals in addition to birds. We plan to identify those from the audio data, even if I just mentioned birds in my presentation. And then Vanessa Mata was asking that wouldn't it make sense to use or improve the available tools for bird sound ID like BirdNet? And indeed, as Petteri already answered, we are collaborating with BirdNet and with Cornell with the Macalau library, so that we don't try to invent the wheel ourselves, but we utilize what has already been done, and then we try to improve on that. So that's a very important point. There was a question by Susan Carrier about how about adding a solar panel to the battery. I think this was maybe related to the cyclone sampler especially. How about either Dee or Bess, do you have a view about this topic?
Dee, I think you have more experience with these, yeah? You're muted.
I was reading the chat and missed the question.
So how about solar panels for the cyclone sampler batteries? Work together with a car battery and charge a car battery.
I know some other teams have done it, but we haven't. So I know for sure that the Vienna team has it. So I don't know I don't have the specs on hand, but I can look them up.
Yeah, actually, I see that now that there has been answers on this in the chat. That Ricardo, for example, from Spain, tells that they have added a solar panel to the roof of the cyclone sampler with a long-lasting battery and the timer. So it's interesting to see about your experience on how well that is working. But it sounds like a good idea in places where it's where it's feasible. There is question by Shankar Nair. Do you recommend to consider the wind direction for audio recording? And you remember that I showed in my presentation the global temperature map. We have actually already downloaded global data for temperature, precipitation, and wind, daily data for the last 10-year period, and we will keep on downloading this data also as the project goes on. So we actually do have data on the wind speed and wind direction for each of your sites at the time when you do the sampling. So for this reason, we didn't send you equipment to make the weather measurements, because there is always, it would make extra work for you. And also there is always some differences in how exactly people do the measurements. So that's why we thought that it's best to utilize the global databases, which are pretty good resolution at the moment for these kind of measurements. So we do have the wind speed and direction data, and we are looking at what that means. For example, one interesting question is that in the Arctic sites, some of you do spore sampling also in the winter where nothing is produced at the moment. So that's to capture long distance dispersal. So these kind of data are very interesting to combine with the knowledge about the prevailing wind directions. Do we see spores coming from those areas where the wind is coming from? There are a lot of questions flowing in. One question that Luke was asking was that even the better trail cameras last just 6 months or so in tropical rainforest as humidity destroys them, even if you use silica and so on and so on. Should we replace cameras as they go down? If so, who will fund this? This is a very good question. So we do have some backup funds to replace some of the equipment, because it's for sure that some will be lost or destroyed during the course of the project. We are not prepared to replace your cameras about 10 times during the project, which would be needed if they really are destroyed every 6 months. So this is something that we have to think about and discuss with you. But of course, it would be great to have the cameras operational during the whole project. Anne is asking about any advice on malaise traps installation in their desert site, which is extremely windy. I wonder if Tomas would have a point of view on this.
Yes, I just wrote the advice that the traps in themselves, they are super durable and will stand almost anything. I understand that if it's like pure sand, then the trick is really in using as many anchors as possible. But it's unlikely that the actual trap breaks, but it can go flying.
Ricardo was asking that, do you plan to copy all our questions in a question that answers Lifeplan website? And I think that would be a very good idea, because clearly, at least I cannot even read the questions as they are popping up, so that we could check that nothing is missed. What do you say, Bess? Is this possible to do?
Yeah, I'll post this chat on the website. It'll be a text file.
Excellent. So Oscar Khan from Pakistan is asking about what are the benefits to basically to the country. The country's biodiversity director is asking about what are the benefits to the country through participation in the Lifeplan by sharing genetic resources with Lifeplan. And I think this is something that is defined in the data sharing and benefit sharing agreement. So Bess, do you want to summarize those benefits?
Yeah, the ABS provisions, basically for this project, it is that you get information on the species that are present at your site, and you get access to, so we're going to have some training sessions on the methods involved, but that's basically what we mostly on a country level, what a country gets in return for this, and they keep control of their genetic material in the sense that they get to decide what happens to it according to their country's individual ABS regulations.
Thanks. Nicolas Lecomte was asking that could we develop a strategy to measure detection probability for the acoustic recorders? And this is actually a very good question. We have plans to this direction, not only for the audio recorders, but for all the methods, that we plan to do kind of an additional experiments that are aimed to better understand the sampling process itself. For example, if you talk about acoustic recorders, like from what radius are they actually capturing the birds, and how that depends on the bird species or the frequencies calling and all of that. So we do plan to actually hire PhD students, postdocs to do these kind of experiments, because doing these kind of experiments can be quite tedious. For example, you can imagine a dense grid of recorders placed in, let's say, in a forest near Uppsala, for example, where we would synchronize them very well, and so that we can then listen which of the recorders simultaneously capture the bird species as they are vocalizing either the natural birds or some playbacks. And these kind of data then gives us the possibility to parameterize an observation model for the audio data. Similar experiments we plan to do also with the other methods. So if some of you are interested in collaborating in this kind of a more involved methods development or additional experiments, then you are most welcome to take part to this. Thomas Meissner was asking whether it's planned that all teams change the sampling site again this September to return to the original schedule. And this is actually a very good and relevant question, because the schedule has not been exactly as we planned from the beginning, of course, partially due to the COVID. So Bess, do you want to answer this?
So the idea is still that everybody will change at the same time, but we haven't quite decided what that date would be yet, because we want to get to a point where enough sites have started sampling, and then decide, okay, we start from now and we go a year, and then we all switch together. So I agree, we do need to decide this very soon, because of course you all need to make a lot of arrangements logistically when you switch sites. We're going to try and make this decision as soon as we can.
Yeah, a related question, that as the sampling is starting differently for every site, we assume sampling for a whole year, but how will this affect season effects while comparing among the sites? And in the transient, in the beginning, in the transient, before everybody has started the sampling, it's kind of unavoidable that some people will sample longer than others before we synchronize. In the end, we want to synchronize. Still, this data before everybody's sampling is not going to be wasted. So I think the best we can do is to account for the season in the phase when we analyze the data. For example, I showed you the some pilot results from the GSSP project. There we used season as one part of the model to understand the seasonality of the fungi.
There's a few questions here about the ethanol used in the Malaise sampling. And Tomas, I always send these ethanol questions to you because you're the booze expert. So can you go over like, what are the requirements and why we don't like denatured alcohol?
Yes, now I didn't read the original question, but if the specific question is what you just said, then the fact is that quite a few of the different alcohols used for and other substances used for denaturing, they do hamper DNA quality, and some of them actually wreck DNA quality. So you absolutely do not want to use denatured ethanol. And the other thing that people tend to ask us is whether you can use 70% ethanol because they are so much nicer for a taxonomist when they don't get hard and brittle, but you can't because DNA preservation suffers from that added water. So do use full strength ethanol always, is the advice.
Can I have a follow-up question?
Please.
There's actually so many types that are denatured, and there's also different language used by each suppliers. Can we get a list of what you mean? And also that kind of ethanol is turning out to be difficult to get because of COVID. But we will need a list of exactly what you mean by not denatured and full strength, reagent strength, or ethyl methanol and everything. So each company has different names. And so we would like you to go around the world and see what Fisher is talking about and what, please, so that when we order something, it will be quite the process to order things, especially in the areas where there's no road. You cannot fly that product. It cannot go on airplanes. And so I don't know how Cambridge Bay will get it, for example. Anyway,
yes, but this is because there are the different products, and quite a few of them are unsuitable. The advice is to simply so the content should say only ethanol, which is ethyl alcohol, at C2H5OH, and nothing else. And I mean, it's 96% if it's pure, and you only get from 96 to 99 by adding salts to get rid of even more water. But the content should be simply ethanol, nothing else. That's because by that recommendation, we will avoid like having to deal with all kinds of cocktails, of which many are unsuitable.
Yeah, for Suzanne's case, we can actually easily find out because she's in Canada, and we can ask, well, what it's sold under in Canada. So we are actually running out of time at this very minute, and unfortunately we didn't get to all the questions. I wanted to pick out one more, which was from Masai Shihara, which was, can she mention Lifeplan on her university website and stuff? Yes, please go ahead. This is all as public as you like and open. So please feel free. But we're going to have to finish up this session, and please keep asking us more things in the Slack and on email and other ways. And if we get lots of feedback, maybe we'll do this another time. So I wanted to thank everybody, especially Team Perth and Termoli again, for those really good videos. It has been a very hard year to start a global collaboration project. The pandemic created a lot of delays with universities being in shutdown, and and getting equipment because supply chains were disrupted all over the world. But even though we have a long way still to go, we have already come a long way. And thank you very much for all your hard work and patience. Keep pushing us. We're here, we want to help you, and stay in touch. Thank you. Bye-bye.
Thanks very much, everybody. Bye-bye.
Thank you.